Okay, whenever you're ready.
Hello, hi, my name is Eugene Gonzalez Lopez. I have always often wondered why is it important to be involved in biomedical research. Once I got involved in research, I understand the answer became surprisingly clear. Our health, the health of our planet, the health of our children is directly related to biomedical research. Either you, a loved one, or one of your friends or family members will have a devastating disorder sometime in your life. Today I would like to share with you what is biomedical research, what role does animals play in biomedical research, as well as give you an example of my own research. Okay, so what is biomedical research? Webster's dictionary states biomedical research essentially means bio is life or living organism, medical is the art of healing or healing sciences, and research is essentially the inquiry of. So if we put this all together, biomedical research is essentially the search for ways to heal living organisms. Why is it important? Biomedical research has improved our lives tremendously. Pretty much everything we own at home, if it wasn't for animal testing or some sort of research involved, those could have affected our health in adverse ways. Okay. Due to medical research, life expectancies has dramatically increased. In 1900, it was average that we lived to about 50 years old. In 2007, it was determined that the average is about 70 years, and the model suggests that in 10 years from now, the age will increase to 80 years old. This was acquired from the World Health Organization. Now that we know what biomedical research is, I'd like to explain the role that animals have in it. Why do we use animals in biomedical research? Animals are surprisingly very similar to us, with the exception of our physical appearance. When it comes to cells, neurons, organs, they are surprisingly very similar. In some states, I'll get back to that, sorry. Biomedical research has used animals to treat many disorders. If you look in the slide behind me, that is a combination of the most common disorders that have been used treating with animals. If you add all these numbers together, that is more than half the population of the United States of America. And this slide was recovered from the World Health Organization. There are two types of animal research. There are in vivo and in vitro studies. In vivo essentially means, it derives from the Latin word meaning within the life. Basically, in vivo means using the live animal. In vitro means is also from the Latin word saying within the glass, which means that we actually isolate what we want from the animal and we do testing on that. An example of in vitro studies within the glass are cell cultures. Behind me are epithelial cells on the left and stem cells on the right. And these cells were isolated from newborn pigs, and they were used to grow different organs. So these are examples of in vitro studies that they'll isolate the cells, put it in a test tube, grow them, feeding them media, and then put those into a live animal, which will then turn them into in vivo studies where they will grow new organs. 71% of the awards given in biomedical research from the Nobel Prize Committee is given with research in the animal studies. The last two very important Nobel Prize given in medicine is from Bishop Vargas, who basically studied the cellular origins of different retroviruses in chickens, and this was able to be used to put antibodies into the chickens before we cooked them so that we wouldn't get sick from processes that the animals have. Another good example is in 1966, Hug and Rauch studied tumor-induced viruses on rats, rabbits, and hens, and this was very important to finding ways to do our basic cancer research, to find ways to, when the cancers or tumors are in our brains, to actually make them know where they spread and how to degrade them or break them down. Okay, now that we know how animal research is used in the biomedical sciences, I'd like to talk to you about my research. The research I do is actually in the next building, is in the North Hall. I work in the forensic toxicology lab, and I perform research on rat cells called PC12 cells, which are used as analogous for human nerve cells. Nerve cells basically control different muscle coordination. Basically, me telling my hand to contract is using PC12 cells or neuronal cells. And we found that different pesticides that are used to pesticides or fungicides that are used on our fruits to prevent animals from eating them can actually cause Parkinson's disease. And so my research, we try to figure out how that happens. If you look behind me, these are called my actual slides from one of my researches, from one of my projects. The top layer are controls, which means that we haven't affected them with any pesticide. And if you see, the top layer are a lot brighter, they're more complex. And if you look on the bottom, those are the different pesticides. The DDC, MB, and MZ are all different organic phosphate pesticide that we use. If you look in all the ones on the bottom, you see a holification, which means a big hole in the cell. That means the cells are actually destroying. And with the MZ, you see a lot of cell death. So this basically means these are actually affecting our cells dramatically. So we actually trying to figure out a way on how to minimize this effect. Okay. In closing, I would like to say that now that we know what biomedical research is, we know how animals are related to biomedical research, and we also have an example of my own personal research. I'd like to end this speech with a quote from the United States former Surgeon General, Everett Krupp, who said that virtually every major medical advance for both humans and animals have been achieved through biomedical research using animals. Thank you very much.